Thank you very much, Anthony. Thank you all for coming. A number of years ago, Francis Crick, the co-discoverer of the structure of DNA, published a book called The Astonishing Hypothesis. The subtitle was The Scientific Search for the Soul. The astonishing hypothesis referring to the idea that all of our thoughts, feelings, perceptions, emotions, and decisions consist of physiological activity in this organ, the brain. This actually happens to be my brain. I volunteered in an experiment in functional neuroimaging, and as a souvenir, I got to take home a picture of my brain. And I must say, I was astonished to learn that I had a brain that was exactly the same as the one that I had been teaching about all these years. I don't know what was so surprising about that, but there was something strangely validating about seeing all of those parts neatly laid out. But looking at this organ, one would not immediately guess that all of the richness of mental life, of intelligence, of categorization, of language, of planning, of emotion, could arise from the structure of this 3 pounds of flesh. Now, many of you are familiar with widely popularized studies from functional neuroimaging showing that different parts of the brain can be associated with different psychological functions, validating an even older study from neuropsychology showing that if a person loses a part of the brain from stroke or injury, they can lose a psychological function, and from both sets of studies, we know that vision originates in activity here in the back of the brain. Language is concentrated in an area in the left hemisphere toward the front. Planning and decision-making at the very tip, the poles of the frontal lobes, and so on. But just knowing that a part of the brain accomplishes a particular psychological function doesn't tell you that much more than knowing that the whole brain accomplishes all of consciousness and intelligence and the phenomena of psychology. So in this lecture, I'm going to try to bridge the gap between phenomena that we can experience. I will lead you through some visual and tactile phenomena and show how they can be explained by what we know about properties of the nervous system. But first, a little bit of the rationale for the astonishing hypothesis to begin with. Facts that we have known for quite some time that establish that however astonishing the hypothesis may be, there is enormous evidence that it is true. We know that stimulating the brain with an electrical current while the brain is exposed during neurosurgery can lead the patient to have a vivid, lifelike experience, indistinguishable from reality. We also know that during the course of thought and perception, there are patterns of naturally occurring activity in the brain. We know that the brain is a chemical device, as we see from the effects on mood and alertness and the ability to produce hallucinations from taking psychoactive drugs. We know, as I mentioned before, that when part of the brain is damaged due to a stroke, a part of the person no longer functions. A person who has suffered a lesion in the brain may lose the ability to produce an articulate sentence or to empathize with the feelings of others or to recognize a face or object or to make a concerted plan. We know that surgery, another physical manipulation, can affect the psyche, perhaps most dramatically in the famous split brain surgery administered as a treatment for epilepsy, in which the two cerebral hemispheres are separated by severing the corpus callosum, the band of fibers that connects them, which results in independent consciousnesses existing in each of the disconnected hemispheres, as if it were possible to bisect the soul with a knife. We have every reason to believe that the life and death of the body is coextensive with the life and death of the soul. That is, when the physiological activity of the brain ceases, the person goes out of existence. And we have reason to believe this because despite massive efforts, no one has been able to communicate with the souls of the dead, despite enormous efforts. We also know that the brain has a breathtaking complexity that is fully commensurate with the breathtaking complexity of human thought and experience. If we opened up the head and found that it was filled with a homogeneous spam-like substance, we would have reason to wonder how it could be physically possible for the neural substrate to support all of the wonders of human perception and thought. In fact, we know that the brain comprises some 100 billion neurons, or brain cells, interconnected by some 100 trillion connections, or synapses. If one were even to ponder the combinatorial combinations of neurons that are interconnected, one can see that in terms of sheer physical capability, there's plenty of room in the brain for the complexity that we see in human thought. Just to give you a little feel for the complexity that we're talking about, this is a reconstruction of one cortical column. A column is a little plug of brain tissue on the surface of the brain. This one is 1 cubic millimeter. It comprises 10,000 neurons. Actually, Dale, maybe we'll try the trick of turning off the light, just so you can, first of all, we'll see if it works. And I don't know how well you can see the spectacular density of interconnections. But that's what we're talking about when we talk about the brain, and that is just 1 cubic millimeter of your brain. Multiply it by the size of the brain, and you begin to appreciate the complexity of the system we're talking about. Okay, we can, thank you very much. In order to bridge the gap, in order to go from simply saying that this chunk of brain is associated with this psychological function, in order to really explain away the miracle, explain how it is that brain tissue can result in thought, intelligence, perception, we have to crank up the microscope to the level of neurons and neural computation, because that is the level of analysis in which the ultimate explanation for our thoughts are to be found. That is, how does neural tissue make us smart? What is it about brain tissue, unlike, say, the tissue of the liver or the kidney, that results in intelligence? And how does neural tissue account for the contents of consciousness? And that's what I will talk about this evening. Our hero is this cell over here, a neuron or brain cell. This one is cultured on a piece of silicon. A neuron is a cell, so like all the cells in your body, it has a nucleus where the chromosomes containing the DNA are kept. It is filled with cytoplasm, the biochemical machinery of the cell that keeps it alive and underlies its metabolism. But unlike other cells in the body, the neurons have a particular shape. They have a network of input fibers called dendrites, from the Greek word for tree. Then they have, a typical neuron will have one long output fiber called the axon. The axon at its very end branches into these little finger-like structures called axon terminals. Many neurons are also coated with white fatty cells, which collectively form a myelin sheath. That is the white in white matter. Those are the connections between one part of the brain and another. The outer surface of the brain, where the actual circuitry resides, is called the gray matter, and it's composed of of dendrites and cell bodies, and without that fatty covering, it looks gray. This is a picture of a real neuron. This is with a special fluorescent stain. This comes from the structure in the brain of a mouse. But actually, I think we'll kill the lights again. Thank you very much. All of the orange and white fibers that you are seeing consist of one neuron, a single brain cell. Here is the cell body, the soma, where you can see the nucleus. All of these orange tendrils are the dendrites, or the inputs. All of these yellow fibers are the branches of the cell's axon. So again, just to give you a feel for the complexity that we are talking about, take one of these with all of its extensions, multiply by 100 billion, and that's what we're going to be talking about. The transmission of a signal along an axon is often described as an electrical signal, but it's crucial to realize that what it actually consists of. One thing, it took many years of studying the brain to realize that neural transmission does not consist of the flow of a fluid through a pipe, even though a neuron is a hollow tube filled with fluid, but the fluid does not move. Nor is it the transmission of electrical current along a conducting wire, like the copper wire that allows your light to be illuminated. That is, we're not talking about the flow of electrons along a wire. And indeed, neural transmission is quite a bit slower than electrical transmission along a wire. The typical speed of a neuron is on the order of between 2 and 200 miles per hour. So 2 miles an hour is walking speed, 200 miles an hour would be a kind of slow racing car, compared to the speed of electricity, which is on the order of 670 million miles per hour. Also, mental processes unfold at a rate that is glacial compared to your smartphone, on the order of tens of milliseconds, that is hundredths of a second, a millisecond being a thousandth of a second. Electrical processes take place in the order of nanoseconds, billionths of a second. When you shop for a laptop computer, you'll look at the speed of the CPU. That's measured in gigahertz, billions of cycles per second. The brain operates more on the level of hundreds or thousands of cycles per second. So it's slow. A final difference is that despite the convenient analogy of a neuron to an electrical wire, in a neuron the energy is in the axon. It is not conducted by the axon. It's not like there's an energy source and that the neuron passively sends it along its length. The energy that powers it is in the axon itself. It's more like lighting a trail of gunpowder. So this is a close-up of the cell membrane, the membrane that separates the inside of the neuron, the intracellular fluid, from the outside, the extracellular fluid in which the neuron sits. Can you see this all right, or do we need to darken the lights? So, okay? Yeah. Now, a way of thinking about the chemistry of a neuron is a bit like a bag of chicken soup floating in salt water. Although low sodium chicken soup salted with potassium chloride instead of sodium chloride. So in the extracellular fluid, outside of the neuron, you have dissolved salt. Salt is sodium chloride. When you dissolve salt, it breaks the sodium chloride into the sodium ions, which are these positively charged ions floating around, and the chloride ions, which are these negatively charged ions. Inside the cell, you have dissolved potassium chloride, in a sense, which consists of potassium ions, also positively charged, chloride ions, also negatively charged, but also lots of big globs of protein, which are also negatively charged. That's why I likened it to chicken soup. So the inside of a neuron is different from the outside of the neuron. Because you've got all of these extra negative ions inside the neuron compared to outside of the neuron has a negative electrical charge with respect to its surroundings. One other crucial bit of microanatomy that is necessary to understand how neurons function is these structures here that are kind of like pores in the membrane, but they're active pores. They're not holes. They have gates that can open or close. This is called a sodium channel. When the gate in the sodium channel is closed, sodium ions stay outside the neuron where they belong. However, that gate can open, allowing sodium ions to diffuse in. There's also a potassium channel, which when open allows potassium ions to diffuse out. Now, I have not said anything about what opens or closes these channels, these gates. You're going to have to take my word for it that because of the complex molecular structure, they will open or close depending on the charge of the membrane surrounding them. In particular, if a patch of the membrane surrounding a channel switches from negative in charge to positive in charge, the sodium channel will open. And as a result, the flip in the electrical charge will travel along the length of the membrane. So let me just show you that schematically. When a sodium channel opens, Here the diagram says plus atoms, that's really positively charged ions rush in. Then to rectify that imbalance of charges, the potassium ions move back out so that restoring the charge of minus 70 millivolts in that section of the membrane. However, that entire process causes the sodium channels in the adjacent patch of membrane to open. Now sodium ions pour in over here. Quickly, potassium ions pour out to restore the charge, but that just triggers the whole process in the adjacent stretch of the membrane, and as a result, the flip in electrical polarity moves along the length of the axon. One way of thinking about it is if I were to take a voltmeter. This is a gadget you can get at a hardware store or an electrical supply shop with two leads, positive and negative. Here they are attached to a battery that registers the voltage. A neuron is a bit like a battery in that if you were to impale the neuron with an electrode on the inside and an electrode on the outside, then you would get that charge of minus 70 millivolts because of all of the negatively charged ions and molecules that are floating around inside the neuron. Then when the sodium ions pour in, the charge flips to positive because sodium ions are positively charged. When the potassium ions exit the cell to compensate, the potential of the inside with respect to the outside gets restored to the resting rate and it becomes negative again. However, if you were to measure this change along different positions of the membrane, you'd see that that flip in charge travels along the length of the axon. That is, if it were to start over here, then it would move along the length of the axon all the way to the very tips of the axon terminals. Now, a crucial change happens when this flip in electrical polarity reaches the axon terminals, where the axon terminal of one neuron meets the dendrites of the next neuron in the circuit. That structure is called a synapse. Synapse is the junction or connection between one neuron and another. Here we have a synapse. Here we have it blown up schematically. So we have our neural impulse which travels along the length of the axon until it then reaches all of the axon terminals that form synapses on the dendrites of the next cell down the line. This is the axon terminal blown up, and the axon terminal contains a number of little baggies filled with chemical which are called synaptic vesicles. They are little molecular structures, so you can think of just as little bags of chemicals. The chemicals inside them are called neurotransmitters. And through a bit of molecular wizardry that I will not explain, when the neural impulse reaches a synaptic terminal, it causes the synaptic vesicles to fuse with the membrane at the end of the axon terminal. That means that it is now open to the exterior of the neuron, and it kind of dumps its contents into the cleft between the first neuron, the presynaptic neuron, and the second neuron, the postsynaptic neuron. So these chemicals are kind of barfed out into this gap between one neuron and the other, and they diffuse across. This is a blow-up of the blow-up. So here is a synaptic vesicle. It has fused with the membrane, which means that it is now open to the world. The neurotransmitter molecules diffuse across. Here we've got fluid between the presynaptic membrane and the postsynaptic membrane. Now, one more bit of molecular gadgetry. The postsynaptic membrane is studded with a complex molecule called a binding site. And what this consists of is another channel, that is the gate can open, allowing sodium ions to flow, only if a neurotransmitter molecule fits into the binding site. Sorry, this is called a receptor, not a binding site. The binding site is the little slot into which a neurotransmitter molecule can fit like a key into a lock. And when the neurotransmitter molecule, suffusing across the cleft between the presynaptic membrane and the postsynaptic membrane, fits into its designated slot, then the gate in the receptor opens, allowing sodium ions to diffuse through, replicating the process in the next neuron downstream. Now the whole process begins again, and the impulse can now travel along the second neuron. So the crucial thing to remember is that although the transmission of a signal in a single neuron is electrical, that is the flip of polarity along the length of the axon, the transmission of signals from one neuron to another is chemical. The nature of the transmission shifts from electrical to chemical. And the neurotransmitter molecules, because they are particular kinds of molecules, are allow pharmaceutical manipulation. Here are a few of the neurotransmitters that are used in different circuits of the brain. There's norepinephrine, also called noradrenaline, which is related to the adrenaline that your adrenal gland produces and is involved in the brain in alertness and activation. There's acetylcholine, which allows neurons to communicate and contract your muscles. There is dopamine, which is involved in a number of circuits, including ones that are related to wanting, craving, desire, and addiction. There's serotonin, associated with a feeling of calm or well-being. Glutamate, the same amino acid that is in MSG, monosodium glutamate, and that is the main excitatory neurotransmitter in your gray matter, your cerebral cortex. GABA, G-aminobutyric acid, is another neurotransmitter in your gray matter, your cerebral cortex. The general point, though, that I just want you to keep in mind for now, since there's a lot of material coming at you quite quickly, is the fact that the transmission from one neuron to another is chemical rather than electrical is why there is such a thing as psychoactive drugs. They enhance or interfere with the diffusion of chemicals across that gap and therefore can affect your mental functioning. Caffeine will make you more alert, and LSD will make you hallucinate, and Prozac will change your mood, and so on. One crucial property of synapses is that they come in two different opposite kinds. I've so far talked about excitatory synapses, symbolized here as little e's, which means that if the input neuron is active, then that will increase the probability that the output neuron will fire. Now, if that's all your brain contained, there would be two big problems. One would be, since ultimately every neuron is connected to every other neuron, if all you had were excitatory synapses, one neuron would excite another, which would excite another, which would excite another, which would eventually would loop back to the first, and your whole brain would kind of blow up in one huge seizure. The other problem is that you can't really do any interesting computation if all you have are excitatory synapses, for reasons that I will soon explain. And not surprisingly, the brain has a second kind of synapse called inhibitory synapses, here indicated by the eyes, where if the input neuron is active, that decreases the likelihood that the output neuron will fire. So the presynaptic neuron can either increase or decrease the rate of firing of the output neuron, depending on whether we're talking about an excitatory or an inhibitory synapse. Okay, so what gets this process started? Obviously, your brain is not just a random neural activity generator, but something has got to make your patterns of activity in your brain actually register something in the world. And there is a particular kind of neuron that is sometimes called a feature detector, where that neuron will respond to particular patterns coming in from the sense organs, such as the eyes. Now, now I think we didn't test the sound, but I hope we have some. So a feature detector is a neuron that responds to patterns in the input. I'm going to show you now a recreation of one of the greatest discoveries in 20th century neuroscience by the neurobiologist David Hubel and Torsten Wiesel. They won a Nobel Prize for the little demonstration that I'm going to show you right now. David Hubel is explaining how they came upon this discovery almost by accident. And the sound should be working, right? And it's probably a good idea to turn off the lights as well.
When we started working, Torsten and I, in the late 50s, we set up our first experiments, and they didn't go well because at the beginning we couldn't make the cells fire at all. We'd shine lights all over the screen, and nothing seemed to work. And rather by accident, one day, we were shining small spots, either white spots or black spots, onto the screen, and we found that the black dot seemed to be working in a way that at first we couldn't understand until we found that it was the process of slipping the piece of glass into the projector, which swept a line, a very faint, precise, narrow line across the retina. And every time we did that, we'd get a response. And even more than that, the line produced a response as it swept across the screen in one direction, but not in the reverse direction. Of course, that could have simply been an oddball cell. We didn't know whether we'd ever find another such cell, but after some weeks or months, it became pretty clear that most of the cells that we encountered in the visual cortex demanded just that kind of stimulus. Although from one cell to the next, the orientation varied, and a number of other things differed. The line was the important thing.
Okay, so these were experiments done on cats, and the sound that you were hearing, consists of taking the leads of a microelectrode that impales a single cell in the visual cortex of the cat, amplifying that very tiny electrical signal, the one that goes from minus 70 millivolts to plus 20 millivolts, feeding it through a speaker so that every one of those action potentials is a pop, and when you hear pop, 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 that each one of those is a neural firing. When you hear brrr, brrr, that is the neuron kind of firing its head off. And so when you heard the brrr sound, that consisted of a single neuron in the cat's visual cortex responding to a moving diagonal line. Now, the reason that that is significant is that the input to the brain does not consist of lines. You've got rods and cones in your eyes. Each one responds to a little point. You kind of get a pointillistic picture of the world that is sent to your brain. If there are cells in the brain that are, in effect, line detectors, there must be some circuitry that converts the point-like stimulation into the pattern consisting of a line. Hubel and Wiesel speculated that the neurons are wired in an arrangement more or less like this, that you have this is a patch of your retina, or the cat's retina in this particular case. Here are a bunch of cones, and they are wired together so that all of the cones here and here and here feed into a particular neuron in the brain itself, so that only if you have an entire array of cones that are stimulated simultaneously will there be enough activation that will cause a cell in your brain to fire as a result. So this is the very first step of going from raw sensation to something that you might want to call a pattern or object. And the general way in which scattered patterns of input can be transformed into some coherent output that stands for something in the world, in this case something as simple as a line, is the subject matter of the branch of neuroscience sometimes called neural computation or neural networks. And just as you can go from the pointillistic activity of cones in your eyeball to lines, you can go from lines to more complicated concepts and build detectors in the brain, so to speak, that respond to more complex concepts. There's, and in fact, what I consider one of the great discoveries in the history of thought, in the 1940s, two neurologists named Walter McCulloch and Roger Pitts showed how you could actually compute logical functions, the kind that go all the way back to Aristotle, if neurons were wired together in particular ways. Now, I think a lot of logicians would say, you give me the ability to compute logical functions, I will give you all of intelligence. So let me just give you a simple example of that. Let's take a fairly complex concept, not something as simple as a line, but the concept of kosher from the Jewish dietary laws, which is defined more or less as follows. Something is kosher if, an animal is kosher, if it chews its cud and has cloven hooves, or it has fins and has scales. So the reason that pork is not kosher is that pigs don't chew their cud, even though they have cloven hooves. The reason that trout is kosher, but lobsters and clams are not, is because lobsters and clams don't have fins and scales. The point just being that a complex concept like kosher can be built out of simple concepts like has fins if you are allowed to connect them with ands and ors and nots. And what McCulloch and Pitts showed is that indeed very simple neural circuits could compute and, or, and not. And here's the crucial bridging insight. Here is a simple neural circuit at the bottom. These are a bunch of input neurons. This is a synapse. This is the output neuron. The little numbers that I've written along the bottom correspond to the strength of the synapses, namely, given that an input is active, how likely is it to increase the firing rate of the output? And synapses can vary in their strengths. The way that neural computation, in its simplest form, is thought to work is, if you take all of the input signals, that is the firing rates, whether they're going or going number of spikes per second, multiply each one by the strength of the synapse, add them up. If they exceed a quantity called the threshold of the output neuron, how much total activation it has to have before it fires or increases its firing rate, that's the way neural computation, that's the elementary capability of neural circuits. So let me just show you how that works for and, or, and not gates. Here is an AND gate. By the way, I should mention that the chips inside your computer, at the very basic elements, consist of a bunch of and, or, and not gates made out of silicon rather than living flesh, but this is how living flesh might do it. So these three neurons can function as an AND gate. What does AND mean? The statement P and Q is true if the statement P is true and the statement Q is true, logically enough. How do you build a neural circuit that thinks that thought or computes that function or does that little eensy-weensy bit of intelligence? Well, here are the two inputs. Let's call this P, let's call this Q. Let's say when it fires, at a rate of 1, we'll say that that corresponds to a belief that the proposition is true. When it fails to fire, that is the rate is 0, we'll call that a belief that the proposition is false. Now imagine that they are connected to the output by weights of 0.4 and 0.4, and that the threshold, the output neuron for P and Q is 0.5. Well, now what happens if the P neuron is firing and the Q neuron is silent, corresponding to the belief that P is true and Q is false? Well, you have 1 times 0.4 plus 0 times 0.4 is 0.4, which is lower than the threshold, the minimum amount needed for the output neuron to fire, and so this circuit will be silent. Likewise, if the first neuron fails to fire, corresponding to a belief that P is false, but the second one does, corresponding to the belief that Q is true, 0 times 0.4 plus 1 times 0.4, 0 plus 0.4 equals 0.4. 0.4 is less than 0.5. Again, not quite enough input to get the output neuron to fire. But now let's say they are both active simultaneously, then corresponding to the two beliefs, P is true and the belief that Q is true, 0.4 times 1 plus 0.4 times 1 is 0.4 plus 0.4 equals 0.8. 0.8 exceeds 0.5. That's enough to arouse this neuron out of its stupor, and so it fires. And so what I have just described is exactly what you need to have a mechanical gadget that implements the logical connector AND. Over here, we've got an OR detector, if you will. Here, all I've done is I've increased the strengths of the two synapses to 0.6 and 0.6. So P or Q, what does that mean? P or Q is true, if P is true, or if Q is true. And sure enough, if one of these neurons is firing and the other one is not, then 1 times 0.6 plus 0 times 0.6 is 0.6. 0.6 exceeds the threshold of the output neuron, which is 0.5, and so either one of the neurons firing in isolation is sufficient to fire the whole circuit. That is isomorphic to what you need to compute the logical function OR. Finally, imagine that you've got a neuron with a threshold of 0. What that means is that the neuron, even if it is not stimulated, is firing spontaneously. It's just sitting around going brr, brr, brr. And you have a single inhibitory neuron. That is, when the input is active, it lowers the firing rate of the output. Well, now if we call the input P and we call the entire circuit not P, sure enough, when the input is on, corresponding to the belief that P is true, the output is off, and vice versa, and that's exactly what you need to implement a not detector. So the three basic physical devices that are needed to compute simple propositional logic are easily buildable out of simple combinations of neurons. One way in which you can bridge the gap, literally bridge the gap from mind to matter. If mind, at least the intelligent component of mind, consists of computing logical functions, we now see how matter, not just matter can do it, but the exact kind of matter that is found inside your head, gray matter to be exact. Now you might object, isn't logic a confining Procrustean framework for understanding human thought? Humans are not logicians, they're not computers. A lot of our thoughts are squishy and graded and analog, and that is certainly true. So for example, take the concept of vegetable. Unlike the concept of kosher, which has very strict necessary and sufficient conditions. The rabbis will assure you of that. Something either is kosher or it isn't. There are no shades of gray. What is a vegetable? Is parsley a vegetable? Is garlic a vegetable? I mean, celery sure is a vegetable. Everyone agrees. But vegetable is a fuzzy concept. Things can have more or less degrees of vegetable-hood, one might say. But it's very easy to wire up a circuit that can aggregate statistical information as well. Imagine you've got your vegetable detector over here, and its input consists of feature detectors for the kinds of characteristics that are typical but not defining of vegetable-hood. So you've got a green detector, a leafy detector, a crunchiness detector, and so on, each one of which has a positive but low synaptic strength. Then there are certain things that just rule out something from being a vegetable. If it gets up and it walks away, it's not a vegetable. And so a detector for mobility would have a strong inhibitory synapse symbolized by a negative coefficient over here. And so you can imagine that the greater this fires, namely the larger number of typical vegetable properties are present, that corresponds to your degree of belief that something is a vegetable. I can't help but add that in the 1980s, when the Reagan administration was slashing budgets for school lunches for poor children, and they were criticized for starving or malnourishing the nation's poor children, one official claimed that actually it does, that the new diet does contain vegetables. When journalists read the fine print, it turned out that he was categorizing ketchup as a vegetable. So ketchup is a very poor example of a vegetable, but it just shows that the concept of vegetable is elastic. Now, this is quite simple. I've just talked about building slightly more complex concepts out of simple ones, but that can be done layer after layer to build up more and more complex neurons corresponding to features. So this is a simple model that could recognize printed words. At the bottom, you've got your feature detectors of the kind that Hubel and Wiesel documented in cats. Here we have a horizontal line detector, a vertical line detector, a diagonal line detector. Here we've got a number of neurons that theoretically could respond to letters. That is, so for example, the letter N would get an excitatory input from a vertical line detector, an excitatory input from a diagonal line detector, and an inhibitory input from a horizontal line detector. And each one of the letter neurons would inhibit all of the others. At the next level up, you could have word detectors, where the word able would get an excitatory connection from an A, an inhibitory connection from N, and so on. And you could build more and more complex concepts. Now, this sounds a bit like a theoretical abstraction, a fantasy, but I'm going to now show you that the brain of a monkey really does have single neurons that respond to quite complex combinations of features. Here we're going to literally listen to the firing of a neuron, not in the primary visual cortex, the first way station for information coming up through the eyeballs, but several connections downstream. In the bottom of the temporal lobes, there are slabs of brain tissue that respond to complex shapes, including faces.
They can record from single neurons in part of the inferior temporal lobe, and these neurons will be activated by faces better than by any other kind of stimulus that they can present. Not only will they be activated by faces, but if the faces are distorted in certain ways, the neurons will not respond.
As we watch, we can hear single cells in the inferior temporal lobe of monkeys reacting to a single monkey face. The cell only fires if the face is at the right angle and is not distorted.
Okay, now, it is not in general true that for every concept we have one neuron. Nonetheless, in the human brain, there are some that it's been discovered by doing an experiment similar to the one that you just saw with a monkey, but not, of course, to satisfy the curiosity of a neurobiologist, but during exploratory surgery for the treatment of epilepsy. But a neuron was found in the brain of one patient that responded only to pictures of Jennifer Aniston. So that particular patient had a Jennifer Aniston detector. Okay, I promised to relate the title of the lecture was From Neurons to Consciousness, and I promised to show you how properties of neural networks that we can detect physiologically can actually explain aspects of your consciousness. Now, this is, I'm going to be making an assumption here which I'm not going to defend, and which itself could be the topic of not one lecture, but many courses, and that is that consciousness consists of patterns of information in neural networks in the brain. And I'm not going to explain how you make the leap from neural activity to the subjective experience of an entity, just to show you how when the information changes due to properties of the wiring of your neural networks, your consciousness changes. And these are three, so far when I've showed you the various neural networks, the vegetable detector, the word detector, the kosher detector, and so on, the and or gates, all of that was theoretical. But now I'm going to show you three properties that we know to be true of many neural networks in the brain that can explain aspects of your conscious experience. And they're called, just to throw out the jargon, I'll explain each one in turn: lateral inhibition, opponent processes, and habituation. So just a quick refresher on how you see. When you are espying a flower, light bounces off the flower, is reflected off the flower, focused by the cornea and lens of your eye, forming a sharp image on the back of your eye, the retina. The retina consists of light-sensitive tissue. That's where the rods and cones are. Here is a blow-up of a patch of your retina. Here are the rods and cones. They form synapses with a couple of layers of living tissue, whose axons then are bundled together to form the optic nerve emanating at the back of your eyeball, which then connects to the visual brain. So the light goes through, actually your retina is installed backwards, so the light actually goes through several layers of living tissue before hitting the rods and cones. They convert light into neural activity, and the neural activity is sent down to your brain. Now, lateral inhibition refers to a particular way that the neurons in your eyeball are interconnected. This was discovered many decades ago, but it's a widespread phenomenon across the animal kingdom and in many circuits of the brain, that instead of there being one-to-one connections, each input to each output, there are branches, so that each input neuron turns on its output neuron, but turns off the neighboring output neurons. So imagine for each one of you that if I stimulated you with a laser beam, you would do two things. You would and let's say I did it for, I hit one of you with a laser beam, you would stand up, you would simultaneously push your neighbors down. Now imagine everyone's doing that at the same time. That's lateral inhibition. So let me just explain in detail how that works. This is a little plug, a little patch on your retina. The R's are receptors, that is the cones that respond to bright colored light. And they're wired in such a way that for each one there is a bullseye, a area in the center of a patch somewhere in your retina, such that the receptors, the cones, excite the output, the ganglion cell. However, each one of those bullseyes is surrounded by a donut where the cones in that donut, through a couple of synapses, inhibit that ganglion cell. Okay? So this wiring diagram that you are now looking at is an example of lateral inhibition. Okay? The center excites the output cell, the surround inhibits the output cell. Now you've got to imagine that your retina is studded with circuits like this one over here. So every one of the connections between your eyeball and your brain, the little point of light is turning on its output, turning off its neighboring outputs. I'm going to show this in a slightly different way. This is a schematic diagram. These are your receptors, your cones. These are the ganglion cells that go to the brain, and the arrows are excitatory synapses, the dots are inhibitory synapses. Here is a simplified, simple version of a lateral inhibition circuit. You can see the input is exciting the ganglion cell. The neighboring cones, the neighboring inputs, are inhibiting the output cell. So what's the point of that? What does that do if you imagine that every single one of the fibers going from your eyeball to your brain is being excited by one point in the retina and inhibited by surrounding points in the retina? Well, here's what happens if you, let's say I were to shine a little spot of light on your retina. The light is plotted over here. This is position along the retina from left to right. This is intensity. So the fact that we've got this square function here simply means that we've got black, 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 black as we go from left to right, white, 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 black, 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 black. You feed it through this array of lateral inhibition circuits, and what is the output going to look like? Well, you can probably figure it out intuitively. If you did the arithmetic, it would be very easy to see that the output would not replicate the input. It would not consist of off, 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 on, off, 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 off. Rather, it would enhance the response at the boundaries so that you have medium, 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 inhibited, excited, medium, 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 excited, inhibited, medium, medium, medium. So you basically have a circuit that converts the input from just stuff that's brighter and stuff that's darker to finding and enhancing the edges. And once you know that that's the way your eye-to-brain connection is wired up, you can explain a number of illusions that have been known by psychologists and philosophers for centuries. And I will quickly run through them: Mach bands, the Herring grid, and simultaneous contrast. Adele, can you hit the lights? Okay, Mach bands, discovered by the great physicist Ernst Mach, consist of the following sensation. Here we have a number of patches going from black to white in various shades of gray. Each one of those rectangles is uniform, but I think you'll agree that they don't look exactly uniform, that for each one, the left-hand edge looks a little brighter and the right-hand edge looks a little darker. little brighter, little darker, little brighter, little darker, little brighter, little darker, and so on. Why is that? Or actually, let me just summarize the pattern of activity. Here's another example of, and you can again see the Mach bands, darker over here, lighter over here. So if this staircase plots the physical intensity hitting your retina, this spiky zippery function shows your perception of subjective lightness. The explanation is as follows. For a ganglion cell that responds to a patch in the middle of a gray area, it is getting excited by the cones that are located over here in your retina. On the other hand, it's also getting inhibited by all of the cones that surround it. So it's kind of a wash. It's getting excited by its own input, getting inhibited by its neighbors, so it's going to have a kind of iffy anemic response. Now compare it to a ganglion cell that gets input from a region close to the boundary. Well, it's going to get excited, as the A1 does. It's going to get inhibited from the cells here, here, and here. On the other hand, since we've got a darker patch over here, there is less input into some of the input neurons that ordinarily inhibit it, so it's getting the same amount of excitation, but less inhibition. That means it fires at a higher rate, and firing at a higher rate translates in your own consciousness as a slightly lighter shade of gray. And that is, and conversely, for a patch in a darker shade of gray, it's not getting a whole lot of input from its own cones, but nor is it getting a whole lot of input from the neighboring cones. On the other hand, a bit of dark patch is getting not much input from its own cones. On the other hand, it's getting a lot of stimulation that from the neighboring patch that is going to be turning down its firing rate, and so this part of the patch will seem darker than usual because its firing rate was never very high to begin with and is further depressed by all of this activity coming from its neighbors, which are turning down its own rate. Here's another perceptual illusion called the herring grid, known since the 19th century, that may also be explained by lateral inhibition. If you look at this for all, you'll probably see a lot of ghostly gray squares in the intersections, yes? Except for the intersection that you're actually looking at. So as your eye flits from intersection to intersection, there should be gray squares everywhere but the intersection you're looking at. Explanation is that for a bit of white in the middle of the city blocks, it's getting stimulated by its own input, it's getting inhibited by its to the left and to the right, but not getting inhibited at the top or the bottom. So it's a fairly strong signal, whereas the ganglion cells that respond to an intersection are getting excited by the cones that they are directly connected to, but they're getting inhibited 4 times, above, below, to the left, and to the right. With all of that inhibition coming from the neighbors, that lowers the firing rate. Lower firing rate equals perception of gray rather than white. Here's another example. You should see faint diagonal gray lines for a similar reason. But the best one of all, this was actually an internet meme that circulated by email a couple of years ago. Here you should see lots of scintillating gray spots, except at the spot that you are actually looking at. Hearing good. Finally, simultaneous contrast. I think this is probably a better illustration. Depending on the way the projector is working and where you're sitting, this gray spot should look darker than that gray spot, even though physically they are the same, as you can see when we connect them or when you swap them. Now that becomes slightly darker, that becomes slightly lighter. That's called simultaneous contrast, and the explanation is that when a patch is surrounded by black, it is not getting inhibited by its neighbors, so it can party and scream its head off. For a patch that is surrounded by a bright surround, all of those neighbors are telling it to pipe down, and so it is perceived as darker than it is in reality. Why is the brain wired up to in this lateral inhibition circuit? It's certainly not to generate Mach bands or cool illusions, but basically it's a way to get the brain to ignore boring stuff, namely what is constant across the entire visual field. and to highlight interesting stuff, namely where an object leaves off and the empty space around it begins. So if you imagine that this picture of a rose is the input to a lateral inhibition circuit, if you imagine the output to actually fill in the boundaries, the lines, that's kind of like what lateral inhibition does. Those of you who play with Photoshop and sharpen your photos are doing something very similar to what the brain does in a lateral inhibition circuit. Okay, two more features of neural circuitry which respond to differences and which in turn can explain aspects of your consciousness, and then I will end. Another feature that is common throughout the nervous system and in many, many animals is called opponent process circuitry. And this is how the brain registers not differences in intensity, that is, black to dark gray to light gray to white, but differences in quality, the difference between red and green, hot and cold. So the way that an opponent process circuit works is you've got two inputs to one cell that detect qualitatively opposite features of the world. So imagine that you've got in your skin a detector for heat and one that detects colder surfaces. And let's say they converge on a neuron so that one is excitatory, the other is inhibitory. And the signal comes out and it is neither a hot detector nor a cold detector. It's a hot versus cold detector. Likewise, imagine that in your eye, you've got a cone. In fact, I'm going to show you this. Here you've got a green cone. It is a cone in your retina that responds best to green light. Here you've got a red cone. It responds to light that is red. And they are wired up so that the ganglion cell, which goes to your brain, gets an excitatory input from the red cone. It gets an inhibitory input from the green cone. And so it detects neither redness per se nor greenness per se, but red versus greenness. That is, when this neuron is excited above its resting rate, you see red. When it is inhibited below its resting rate, you see green. So two different qualitative sensations encoded in the relative rate of a single neuron. Now this is crucial to the next couple of demonstrations. For one thing, it explains a phenomenon called simultaneous color contrast, which is similar to simultaneous lightness contrast, but here, instead of the patch that's surrounded by other material changing in how light or dark it is, it changes in the quality of the perception. That is what color it is. Here's a nice demonstration. Lights, please. So here you have a slightly mauve X, a slightly purplish X, and a slightly yellowish X. Right? Wrong. They're actually the same shade of gray, but when the gray is surrounded by purple, it appears yellow. When it's surrounded by yellow, it appears purple. That is identical to the explanation that I just gave of Mach bands, Herring grids, and simultaneous contrast. If you add in the additional feature of opponent process circuitry, namely neurons that respond to, that when they respond give you a sense of quality rather than quantity, in this case the quality being color. Okay, this is the last fact about the about neural circuitry that I'm going to explain to you this evening. And this is a phenomenon called habituation, which is that if you stimulate a neuron continuously, it will start off going brrrr, but then it'll start to kind of poop out. It'll go brrrr, and it'll go down. It can't sustain the firing rate indefinitely. It's as if the neuron kind of gets tired. This has been known literally for millennia. So if you, on a hot day, you want a nice refreshing dip in the ocean, then you dip in your toes, and to your shock and horror, they are icy cold. However, if you stay in one place with the water covering your toes, eventually that sense of coldness goes away, and it feels as if you are in nice room temperature water. Then you go in a little bit deeper. The water is up to your ankles. Your ankles are now icy cold. If you've got the patience to stay put, the receptors from the skin around your ankles will habituate, and your ankles will no longer feel cold, and that's why you gradually accustom yourself to the water by going in gradually. Okay, add together the phenomenon of opponent process circuitry, which I explained a minute ago, with the phenomenon of habituation that I just explained. Namely, show stimulus A for a long period of time, the A cells will then habituate. Now show a neutral stimulus, that is, equal amounts A and B. So in the case of color, that would be equal amounts of red and green, which means white or gray. In the case of hot and cold, it would be tepid or lukewarm water. If after you've habituated a neuron to one quality of input, you now expose it to a neutral stimulus, because the A cells have been habituated, they are firing below their resting rate, but the B cells, not having been habituated, are ready and rested and are happy to do their thing, then a neutral stimulus will not give equal amounts of input from the two qualitatively different inputs, because one of them has been fatigued, habituated, tired out, while the other one is ready and resting. In fact, the neutral stimulus will now overshoot and will be perceived as the opposite of the one that was habituated. Okay? The input from the B's exceed the input from the A's, not because the world has more B than A, but simply because the A detectors have been habituated, and so you will perceive a neutral stimulus as B. Now, this has been known during the onset of modern philosophy, when the value of skepticism in clearing away false knowledge was being emphasized. Erasmus and other philosophers pointed to the following phenomenon as reason why we should not trust our senses. If you put one finger in cold water, another finger in hot water, allow them both to habituate, now put them both in lukewarm water, room temperature water. The finger that was in the hot water will perceive it as cold. The finger that was in the cold water will perceive it as hot. You can look and you see that both fingers are in the very same water, but one finger is telling you, man, that's hot. The other one's saying, brr, that's cold. Kind of proof not to trust our senses. Explanation, while you were habituating them, in one case you were habituating the cold detectors, so a neutral stimulus is now perceived as hot because the cold detectors had been habituated, vice versa for the heat detectors. But this also explains a phenomenon that some of you may have experienced from the back of cereal boxes or little prizes and toys when you were kids, namely color after effects. So here's an example. What I would like you to do is I'm going to show you a false color Union Jack. Again, we'll turn off the lights. I want you to stare dead center at the Union Jack and trying as hard as you can not to move your eyes. So look right there, try not to move your eyes. It's literally impossible not to move your eyes, but you can prevent them from moving too much. I'm going to let you look there for another few seconds. Let's say 10, 9, 8, 7, 6, 5, 4, 3. When I get to 0, what I'd like you to do is to jump your eyes over to the black dot. 3, 2, 1, 0. And if all worked, you should see a glorious red, white, and blue Union Jack. This being a color after effect caused by the fatigue or habituation of the cells that respond to yellow and green. This is an even better one. Stare at this point over here. This again was an internet meme of a couple of years ago. Okay, all of that color is hallucinated by your brain as an after effect. Okay, you get to see it again. Okay, and as you move your eyes around, you'll see the color disappear. Okay, so this is a relatively new one that I particularly like. Again, keep your eyes fixated at the center, at the plus, so that rotating green dot does not exist. It simply consists of a patch that is not pink. It is gray. However, because, at least if you don't move your eyes, the inputs responding to pink have been habituated, therefore gray, which stimulates equal amounts of green and pink, but because the pink inputs have been habituated, the green inputs will predominate and you hallucinate a revolving green dot. Okay, finally, I'd like you to stare at the same thing. Actually, before I show this, a little bit of explanation. The same logic that works for temperature after effects and for color after effects also works for motion. Now imagine that there are detectors in your eye, as Hubel and Wiesel showed was true of cats. Yes, the lights can come back on. Thank you. That respond to different kinds of motion. So imagine that you have neurons that are up detectors, upward motion, and downward motion, and leftward motion, and rightward, and diagonal up, diagonal down, and so on. In addition, there are motion detectors slightly higher up in the brain that respond to inward motion and respond to outward motion. Now, crucially, because of opponent process circuitry, these motion detectors are arranged in pairs, so that you've got cells that are excited by upward motion detectors and inhibited by downward motion detectors. You've got cells that are excited by inward motion detectors and inhibited by outward motion detectors, and vice versa. So now I'm going to ask you to stare at the center of this rotating disc. Again, try not to move your eyes. Actually, if you could have the lights on halfway. Is that possible? Yeah, that's good. Actually, that's fine. It doesn't have to be dark for this. That's excellent. Doesn't matter what thoughts you're thinking. This is a low-level process that takes place in your primary visual, well, in some of the early visual parts of the brain. So even as you're processing my speech right now, it will not interfere with the habituation that takes place, which is a purely automatic process requiring no attention. And when I count down to 0, what I would simply like you to do is shift your gaze from the center of the spiral just to my nose. Okay? 10, 9, 8, 7, 6, 5, 4, 3, 2, 1, 0. And the chuckling confirms that you have just experienced the great exploding head illusion. By the way, this is, you don't even need a computer-generated display to experience this rather striking illusion. I experienced it once while mountain biking, and it took me a while to figure out what I was experiencing, but I was biking along a trail, came to a clearing. There was a kind of a ravine, a canyon, and the opposite bank seemed to be moving backward, kind of receding, back, kept moving back. It wasn't going anywhere, but it was moving backward. Now this sounds paradoxical because motion is change of position. Psychologically, it's not paradoxical. You can have perception of motion without change of position, and it looked like this opposite cliff was moving backward even though it was not changing its position. And at first I wondered, was I low in oxygen, low in blood sugar? Was I having some LSD flashback? I knew it couldn't be that because I never took LSD, so I needed a more rational explanation. And then I realized that I was experiencing the great exploding head illusion in reverse, that after a half an hour or so of going along a fairly narrow biking trail where the pattern of leaves was constantly fleeing the focal point. It optically, because of the projection, there was a lot of visual texture that was moving outward as I was moving forward. Then, when I got stopped and rested at the edge of the canyon, a stationary surface, namely the opposite canyon wall, was contracting inward, the opposite of the outward expansion whose detectors had been habituated, and geometrically, inward optical flow is the same as recession in depth, just as outward optical flow is the same as looming as you approach something. One other example that you can that you might be able to experience by yourself is called the waterfall illusion. If you stare at a waterfall without moving your eyes, then look at the rocks beside the waterfall, they will appear to be drifting upward and paradoxically not be changing their position, but the explanation is the same. So just to sum up some key points that I've covered in this, what I apologize to be a very dense lecture, is how neural firing works, how synaptic transmission works, in particular the change from a flip in electrical polarity to diffusion of a chemical, how the brain can not just fire, but fire in response to coherent patterns, thanks to feature detectors, computation in neural networks, how you can compute both fuzzy concepts and logically crisp concepts, and then three properties of neural networks with large psychological effects, namely lateral inhibition, opponent process circuitry, and habituation. Thanks very much. Thank you. Thanks very much.
Thank you very much
Questions?
indeed, Steve. Thank you. I think there might be some questions if anybody would like to ask some. If you'd like to ask questions, stick up your hand and a microphone will come to you.
Thank you for a wonderful lecture. James Shaftesbury, I'm a philosopher of science. The most important thing in our world is a perception of reality. Most people seem to fail to have a very good perception of reality, namely a majority of people who will believe in things like religion when all of the evidence suggests that it's nonsense. And what you find is that a very small amount of people who are intellectuals will eschew religion, and most of the majority who are not intellectuals will follow some kind of religion. But we've known for a long time that the left parietal and right parietal lobes of the brain tend to, the left controls our dreams and our volition and our wishes, and the right parietal controls our logic. And when there's right parietal damage, you find that people lose track of their logic, and the patient very often is in denial of something. This is a very well-known neurological syndrome. And it struck me that, is it possible that most of the world could have mental illness and be in denial of the logic that we find all around us? Because when you have meta-analyses, if you examine something with dendrochronology and isotopic analysis and carbon dating, the likelihood of it being wrong when all of those things concur is we use less evidence to hang a man in America or give a man the death penalty. And yet when we have that kind of evidence, how is it that probably a majority of people will be in denial of that?
Yes.
Which leads me to one point that I'm very concerned about in England. And that is, we have some very intelligent people who have postulated aspects of our world between different kinds of people, whether they be males, females, or clades or types, who are genetically and inherently different. And yet when they speak these obvious facts that all of the intelligentsia agree with, they lose their jobs or their persona non grata, or they don't get media time anymore.
What's going on? Why are we susceptible to?
How would you approach the problem that the minority of people are obviously right, but the majority won't accept it? What should we do about that?
Well, I guess the most general answer is that none of us has a brain that can actually put us in touch with reality unaided, with truth. I've just shown you a bunch of gadgetry that responds to things that are essential to our survival, things that prevent us from falling off cliffs and bumping into trees, but that don't actually correspond to any clear description of the world as it is. So we have, for example, circuitry that responds to edges rather than the physical distribution of light, because edges are important. That's where objects leave off and their backgrounds begin. Moreover, they can be fooled, as I did with each one of you, that since our neural circuitry does consist of a mechanism, not a pipeline to the truth, or a window onto objective reality, that is naive realism, we know is false because there are illusions. You can't just trust your senses. That means we need some way of augmenting the faculties that nature gave us to make our beliefs as accurate as possible, which of course is why the early modern philosophers made such a big deal of visual illusions. They said you can't just open your eyes and see the world as it is. Look at all these illusions. And so what we do, what gives some of us confidence that our beliefs are objectively true is that they are part of an institutional infrastructure of testing and criticism and logic and determining which hypotheses withstand criticism and attempts at refutation, which devising exotic arrangements of reality to reflect the way reality is put together, namely experiments. Thanks to this huge network of minds, each one of them making up for deficits of all the others, we like to think that we have a collective grasp on truth that none of us individually can have. But of course it's always fallible, and the religious beliefs, I think, were actually pretty reasonable hypotheses in earlier states of our collective knowledge. Even Richard Dawkins said that before Darwin, he probably would have been a creationist because the cosmic engineer was the only available explanation for the obvious adaptive complexity of living things. I would add that prior to the advent of modern neuroscience, it would have been, I probably would have believed in souls. Because there was no better explanation for phenomena such as that people respond intelligently to their environments. When you dream, some part of you is up and about the world while your body is in bed the whole time. That seems like there must be some part of you that can leave your body. When a person suddenly dies, some kind of animating force no longer seems to be animating the body that was there just a few minutes ago. So the soul hypothesis is not a stupid or irrational belief, nor is creationism if it's the best that you have. Now, of course, we can do better. But doing better in part depends not just on this cumulative weight of evidence and argument, but because few of us can actually reconstruct the scientific observations that justify the theory of natural selection or the theory of neural networks, all of us, at least a little bit, take it on faith in the sense, faith in the following sense, that there is a community that you trust enough because when they have been challenged to defend their beliefs, they can connect them to reality. They have shown that it isn't magic, that they have given reason to trust them because they've convinced you that the mores of their culture, of their scientific culture, are designed so that they will find true things and weed out false things. If you don't have that degree of solidarity and trust, if you think that scientists are all in cahoots with big corporations or are trying to tear down all faith and values and meaning, and you simply refuse to follow through the very long chain of deductions that connects the claims to the experiments, then you'll naturally reject it just because it's not your tribe. For you and me, scientists are our tribe, and we're apt to give them some credibility, indeed some credulousness, knowing that if ever challenged, it would be repaid. But unless you trace that through, it can be all too easy to reject the conclusions of science. And indeed, a number of studies have shown that contrary to the common belief that people believe in falsehoods, such as that vaccines cause autism, or that humans were created 5,000 years ago, or that the burning of fossil fuels has no effect on global temperatures. It turns out that people with those beliefs that we have every reason to believe are false are no more scientifically naive on average than people who have the beliefs that are scientifically justified. You give a test on evolution to a bunch of people who believe in evolution, give it to a bunch of creationists, there's not much difference in how well they score. In fact, often they're equal. Likewise, the typical grasp of climate science among someone who signs on to the scientific consensus is not much better than the adamant deniers. The difference is, and that kind of dashes the hope that simply by increasing the quality of science education, you will lead people to more accurate beliefs. There, in addition, has to be a dissociation of who you think are the good guys and who are the bad guys from who has the best claims to truth. That's what it depends on.
Cheers. Thanks, Professor. Can I congratulate you on such an admirably diplomatic and extended and discursive answer there? I think what you say is spot on, and it shouldn't cause offence to the people who are clearly deluded. But I just want to ask you, about 3 years ago, I discovered from Professor Colin Blakemore that the number of bits per second, as it were, from the retina through the LGNs and so on to the V4 cortex is about 50 for each retina per second, which is clearly not enough to, as it were...
50 what?
50 bits per second.
50 bits per second.
Yeah. That's checkable if you want to contact Colin Blakemore. That's not enough, obviously, for me to paint the visual scene here. Now, I appreciate that I'm saccading, but I'm saccading several times per second, so that would give you, say, 10 bits per saccade. So the question is, do you think, is there any evidence, or if there isn't, I don't know what your opinion would be, that there's oculomotor feedback into the direct reading out from, from the neural, from the retinal ray along the optical nerve tracts to the rest of the brain? Because it seems to me there's not enough information. If you look at it in terms of my attempt to paint a picture with a few bits and end up with a few spots, say black spots on a white canvas, and it's not going to represent the Mona Lisa. Yet I see the Mona Lisa within at most a second or so, and all its gory detail, shall we say. So the question is, there must be some collateral input which is not purely optically mediated, but to do with registration of the mechanical movement of the saccades of the eyeball. In fact, I know there are different orders of saccade going up to about that. I've asked several questions. We just had one, didn't you? I said, what is the mechanism? Do you think there's oculomotor feedback? I'm sure you heard that. Yeah. Okay, yeah. So have you any suggestions, or do you know about this? It was a surprise to me, indeed a shock.
50 bits per second seems a bit low to me, given that there are 1 million ganglion cells coming out of each eye. But it is certainly true that the perception that we have of a richly detailed visual field from edge to edge and top to bottom, we know is something of an illusion, because if something is changed when you are not looking at it, the change goes undetected, a phenomenon known as change blindness, and of course the basis of stage magic. Magicians exploit change blindness and inattention blindness, the fact that you actually aren't registering all of the rich detail of the visual field unless you're looking at it and thinking about it, to do their various changes and substitutions and bends and so on in the part of the visual field that you are not attending to, even though the information is reaching the back of the brain. So the question you raise probably can't be answered without a complete theory of vision from the rods and the cones all the way up to full awareness, but it does involve a number of constructive processes that involve both focal attention that differentially accesses certain parts of the input, the control of the eyeballs, you're referring to saccades, namely your eye jumps from spot to spot and your brain cumulatively develops a sense of the entire visual field, given that anything that you're not directly looking at is literally going to be extremely coarse and fuzzy. In fact, here's just I think a simple illustration that will make the gentleman's point vivid. Hold your hand out just about maybe a foot or so away from the place that you're looking at. Don't look at your fingers. Just keep them in the periphery, and not way over in the periphery, but just a few inches out. Now try to count your fingers without moving your eyes. Surprisingly hard, isn't it? You can't easily see even your fingers a few inches from your line of sight, which shows that your vision outside your fovea is surprisingly coarse, and that the feeling that you have that everywhere, for the entire 180 degrees, you're seeing panoramic cinemascope is an illusion. When you need the information, you saccade over to it, then you get nice, rich information. You move away, then you look back when you need it again. But by processes that I think are not completely understood, we build up a sense of the richness of the visual field from a number of saccades. And then even above that, then there's a cognitive process that tends to be coupled with physical eye movements, but can also be decoupled from them, where you've got an internal spotlight of attention that even when your eyes are fixed, you could attend to different parts of the visual field, keeping the eyes straight ahead. So that combination of processes manages to build a coherent visual world out of these patchy snapshots.
Cheers.
Yes, or wherever the microphone alights.
I can speak loudly.
Okay. Well,
It's slightly off topic because it's a little bit of philosophy, but it's a question that really hits me. A lot of scientists that I read,
Yeah. No, it's on. Hold it closer to your mouth.
Okay.
Yeah.
A lot of scientific readings that I make from people who are not cognitive scientists argue that there's no such thing as free will because of materialism. And then some people I read, like Dennett, say that there's a compatibility. And I kind of feel that with a cognitive scientist here, it'd be interesting to ask the question, do you think that there is something even philosophically that we can qualify as free will?
Yes, the question is, since there are a number of neurobiologists, typically not philosophers, who believe that let's call it the astonishing hypothesis, the fact that all consciousness, action, thought consists of neural activity, which is a physical process, which is the point of this entire lecture, that leaves no room for free will, and therefore free will must be an illusion. And so the question is, would I sign on to that interpretation of the physical basis of mental activity? So I hate to say it depends on what you mean by free will, but I would say that the neurobiologists are correct only in the following sense, that it is not true that every time you make a choice, a miracle occurs. That somehow that there is a soul that by processes that are independent of physical activity in the brain just magically causes the body to reach for the chocolate rather than the vanilla. On the other hand, free will does exist in the sense that decisions are controlled at multiple levels of the nervous system, and there really is a difference between your knee jerking when the doctor hits it with a hammer and my deciding to lift up this tape recorder to make a point. They're physiologically different. They involve different circuits in the brain. They involve many very different complexities of input. They differ also in terms of their degree of determinism, that is the probability that you have an output given a certain amount of input. And so a certain subset of processes that go on in the brain involving what we tend to call the highest parts of the brain, prefrontal cortex, frontal pole, that receive inputs from the rest of the brain, combine them with memory, execute decision rules that are not deterministic. That whole family of activities is what we mean by free will. It is not miraculous, but nor does it not exist. And it certainly, we couldn't really make sense of our social worlds without differentiating between actions that are in some sense chosen and in some sense compelled. Even if chosen does not literally mean that down at the level of neurons there has to be some miracle that occurs. So maybe a crude analogy is our best science tells us that even solid surfaces are mostly empty space. On the other hand, it's a little misleading to say that solidity is a myth, and that therefore I should have no more confidence in standing on this stage than on a puff of smoke. There is a real difference, but at a certain level of analysis, it's not what our intuition might say it is, but the difference nonetheless is real. And so I think it would be going too far to say free will is a myth, although free will may not be what we intuitively feel it to be. That would be my best answer.
Hi. From old work with, not me, but an old book of Penrose, and another book of the quantum brain, I can't remember, they mentioned microtubules and how they could possibly be part of the processing of the neurons themselves. How strong is the evidence that there is even more processing going on at the level of the single neuron?
There could certainly be, I'm sorry to be wandering around, but I'm not, because the voice came out of the speakers, I didn't actually see who was actually producing the sound. Oh, there you are, okay. Better to make eye contact. It is certainly possible that the picture that I gave of neural computation, namely that it takes place at the synapse and that the intelligence of the brain adheres to the pattern of synaptic strengths, is clearly an oversimplification. There's every reason to believe that the propagation of signals within a neuron might be computing certain functions, perhaps, and and OR gates or weighting of information. It's conceivable that physiological processes inside the neuron might be executing intelligent functions. Again, I papered over all of that and just concentrated on the synapse. That having been said, I know of no reason to think that microtubules, a kind of nanomolecular skeleton of the cell, does anything that's particularly interesting, and I don't think there's been any demonstration that any quantum level process has anything to do with any psychological phenomenon, nor is there any mystery that needs appeal to quantum mechanics to explain it. So I think that is not a very promising direction. It would be one thing if we had ESP and could sense things at a distance, and we might want to use entanglement, but since we don't have ESP, there's nothing to explain and no need to invoke quantum weirdness.
Hi, thank you for your lecture. I was hoping to inquire about mental enhancement, because some students and some people, will use drugs such as Ritalin to improve their memory or overweigh their mind functions for certain tasks. I was wondering whether these drugs, whether they have long-term negative effects, or whether as a society we should frown upon their usage the same way that we disagree with the usage of drugs for improving our abilities in sports and in other areas, or is it something that we should embrace and something that people should use to improve themselves and improve their abilities to function as human beings?
So is this kind of an ethical or policy question of whether we ought to encourage drugs that enhance cognitive performance?
Both ethical as well as what the potential risks might be, as well as whether it's actually feasible.
Yeah, probably safe to say that you when it comes to the brain, you can't just do one thing. So that if you ingest a substance that changes the function of the brain, it's probably, if it does some good things, it probably does some things that are not as good. But there's nothing to rule out the possibility that there are compounds that whose benefits outweigh their costs. Many of us here benefit from thrice daily doses of caffeine, which is a psychoactive substance whose benefits generally outweigh its costs, unless you have it too close to bedtime and it keeps you up. So I don't think in principle that there's any ethical argument for drugs that improve performance without side effects that do more harm than good. I don't know enough about studies of the effects of these drugs to know whether we actually have such substances above and beyond caffeine, but it's certainly possible that they'll be developed.
Hi, I'm a teacher, and neuroscience and cognitive science are very trendy in schools at the moment. I'm just wondering what you think the role of those can or should be for educators.
Well, I think they're, I think the people in this room came because they find it an inherently interesting subject. That is, how does a branch of biology relate to our everyday experience? So I'd imagine that it would be an appealing subject to high school students if taught well. It's also important philosophically because it is facts of neuroscience are relevant to ancient controversies such as the mind-body problem, free will, consciousness, ones that have, in fact, we've broached in this very room. There are practical applications such as drugs, both psychoactive and recreational. If you bridge from neuroscience to cognitive science, there are also, I think, salubrious lessons in terms of how to remember material, how to understand material, how to distrust your own senses and your own memory so that you're less likely to make errors, how to deflate your own naive self-confidence in the accuracy of your thought processes. Now that's, of course, shading from neuroscience to psychology, but I do think that set of topics is, if skillfully taught, could be highly valuable at the level of high school.